Hello viewers, welcome to Mythology. In this video, let us begin with mitosis. Mitosis is the cell division in which one parent cell divides into two identical daughter cells. More importantly, that the same normal chromosome number is maintained at each mitotic division. Generally, mitosis occurs in the somatic cells, or we call them as the body cells, where the chromosome number is diploid. But it is not true that they cannot occur in the germ cells. Mitosis can also occur in germ cells during the multiplication phase. Mitosis is completed in two steps: karyokinesis followed by cytokinesis. In this video, our focus will mainly be on karyokinesis. Karyokinesis is the division of the nucleus, and it involves four phases of mitosis, that is prophase, metaphase, anaphase, and telophase. Each of these phases merge into the next phase, thereby making it a continuous process. By and large, the four phases of mitosis are quite similar in all living organisms. The slight differences that exist we will speak in another video. Now I'll be describing the stages of karyokinesis in animal cells. The word karyokinesis has been derived from karyon plus kinesis. The word karyon means nucleus and kinesis is movement. Now let us know whose movement are we speaking of. We are usually speaking of the movement brought about by the spindle fibers for the movement of the main thread-like particles called the chromosomes or the chromatin fibers. Let's begin with the first phase of karyokinesis, what is called as prophase. During prophase, the chromatin fibers condense. This condensation is approximately 1,000 folds, which makes them look thicker and thus visible under a light microscope. There is also the spindle fibers which emerge from the centrosomes. Next, the centrioles, they start migrating apart towards the poles. Then it comes with the nuclear membrane, which is fragmented into vesicles. The nuclear pore complexes also dissociate, and the nuclear lamina depolymerizes. As the chromosomes condense and transcription stops, the nucleolus also disappears. Process of condensation allows the chromosomes to move along the mitotic spindle without getting tangled. There is the kinetochore, which is developed in the centromeric region. Polar microtubules that do not engage chromosomes, they overlap midway between the poles and contribute to the cell elongation. The astral microtubules, which are located near the poles, aid in the spindle orientation, which are required for the regulation of mitosis. All membrane-bound organelles like the Golgi bodies, endoplasmic reticulum, and the nuclear membrane, they tend to disintegrate or dissociate. Prophase is quite a long phase. We can categorize it under three heads, like the early prophase, mid prophase, and late prophase. Late prophase is also referred to as Prometaphase. Few characteristics to distinguish late prophase are the complete disappearance of the nuclear membrane, the membranes of the endoplasmic reticulum, and the Golgi apparatus. Next, the centrioles by this time reach either poles. Fibers that arise from the centrioles are basically of two types. Firstly, some smaller fibers which are found at the poles. These are the smaller astral rays. Secondly, the bigger fibers running across the poles, that is called the spindle fibers. In animal cells, since two asters are formed, so it's called amphiastral. But in plants, there are no centrosome or centrioles. That is why no asters are formed, but spindles are formed with the help of microtubules. The next phase of karyokinesis is called metaphase. The characteristic features of metaphase are as follows. The mitotic spindle is fully developed during this metaphase. The centrosomes face the poles. The chromosomes are lined up midway at the metaphasic plate, or what we call as the imaginary equatorial plane. Each sister chromatid is attached to a spindle fiber originating from the opposite poles. The chromosomes are maximally condensed by the protein complex called condensins. The cohesive proteins still keep the sister chromatids highly attached together. The chromatin spindles are attached to the kinetochores, that is, the fibers which are attached to the chromosomes are called the chromosomal spindle fibers or the discontinuous spindle fibers. There are interpolar fibers which run continuously between the two poles. They are not attached to any chromosome. These are absolutely continuous between the two poles. The next phase of karyokinesis is anaphase. The most important feature of anaphase is that the chromosomal spindle fibers start pulling the chromosomes to each pole, so the centromere splits. This separates the sister chromatids which gets pulled to the opposite poles. Then the cohesive proteins that held the chromatids together, they break down. A fiber appears where the centromere was split. This is called the interzonal fiber. There are forces which are developed here due to the contraction of the chromosomal fibers and the stretching of the interzonal fibers. So the chromosomes get pulled towards either pole. That is, with the splitting of the centromere and the interzonal fibers that appear to divide them, during this stage we can see an alphabetical shape of the chromosomes, like the metacentric chromosomes appear in the shape of K, the submetacentric as L-shaped chromosomes, the acrocentric as J-shaped ones, while the telocentric ones appear in the shape of I. Surrounding the centromere, there are kinetochores. And here in anaphase, the kinetochore spindle fibers, they lengthen, elongating the cell. But in closed mitosis, the nuclear envelope remains intact and the chromosomes migrate to opposite poles of a spindle. This happens within the nucleus of Saccharomyces cerevisiae, Saccharomyces pombe, and allied species. Finally, we move to the last phase of karyokinesis. This is called telophase. The features for telophase are chromosomes arrive at opposite poles. They begin to decondense or loosen out, again relaxing into chromatin configuration. Here, the mitotic spindle breaks down. Mitotic spindles are depolymerized into tubulin monomers. These tubulins assemble cytoskeleton components of each daughter cell that will arise. Telophase appears to be reciprocal of prophase stage in quite a few aspects. Let us know them. Firstly, there is reassembly of nuclear envelope by binding of the vesicles to the chromosomes. Then the fusion of the vesicles forming a double membrane around the chromosomes. This is followed by the reassembly of the nuclear pore complexes. The vesicle fuse first to form membranes around individual chromosomes, which then fuse with each other to form a complete single nucleus. The nucleolus, which had disappeared in late prophase, that too reforms again as the chromosomes uncoil and transcription of ribosomal RNA gene begins. A cleavage furrow that was already formed by now starts deepening. This is the start of cytokinesis. Following karyokinesis, it is cytokinesis that takes over. Cytokinesis is the physical separation of the cytoplasmic components into two daughter cells. The cytokinesis is quite different in plants as compared to animals. This we will discuss in the next video. But who gave us knowledge about the mitotic division and the changes in the chromosomes associated with them? Let us know it in details. It was Sir Walter William. In plants, mitosis was first observed by Edward Adolph Strasburger when he saw the dividing cells in Tridescantia. In animals, this was observed for the first time by Walter Fleming, particularly in the larvae of salamander, which is an amphibian. Sir Walter Fleming had also studied the chromosomes for the first time in the year 1882. Now, let us recapitulate the steps in karyokinesis. The karyokinesis involves the four major steps, that is prophase, followed by metaphase, which is then followed by anaphase, and finally telophase. But the cell doesn't divide into two daughter cells only by the process of karyokinesis. It further goes through cytokinesis to divide into two daughter cells. Thank you for watching. I hope you liked my video. If you liked it, do not forget to give a thumbs up, share it with your friends and subscribe to my channel.